So we've been talking about assessing impact, but I think it's good to think about terms to start with. So contamination and pollution, I think most people may think that is exactly the same, but the definitions tell us the contamination is the presence of a chemical or chemicals above what is supposed to be the background, and actually pollution is the observation of effect. So it's important to bear this in mind. And this is where toxicology comes from. So we can link then the studies of effects in the field by linking the chemical analysis to actual effects-based measures. This is what we can actually probe in an individual, on an organism, and trying to see what actually happened to that organism when it was subjected to maybe increased levels of chemicals, and then we can ascertain if we actually can see or observe any toxic effects. So toxicity is really the study of those effects. So the chemical needs to be present, of course, at the site of action, so I'll talk a little bit more about it, but it needs to be available to the organism and at an effective concentration. And this is toxicology or toxicity. When we talk about environmental toxicology or ecotoxicology, of course, we're talking about those studies in an environmental context. So first, when we normally mention in the context of studies of effects on organisms, we talk about hazard, and then we talk about risk. And of course, the hazard is actually the effects of the chemical. The risk compounds the hazard with the exposure. So you would have hazard if you study in the laboratory and you see that the organisms might have died, for example, but they might not happen at that concentration, but they might not happen in the field because that chemical is not present at that concentration. So the risk is really linking the hazard to the exposure levels. So the chemical needs to be available in the environment. Then we need to understand what happens to the chemical. It's transported somewhere. Does it sink? Does it dissolve? Does it evaporate, etc.? And in this context, it's going to maybe encounter organisms which will be exposed to it. So organisms might be either living in the water column, in the sediments, might be exposed to that. Can they take the chemical in? Is that available to them, or is bound to a chemical in a way, another chemical that is make it not available to the organism? So there is no, exposure is then low. And then, of course, it can be transformations, physical chemical transformations. They might make the chemical more available or less available. And it could be physical processes that lead to the transformation of the chemical, or it could be biological mediated by microorganisms, for example. And of course, then we might have an effect. The effect can go for mortality, direct effect, can be effect via the food chain and indirect toxicity, and you might not observe any effect, but the organism might develop some kind of body burden, which might then be transferred in the food chain. So the organism, when confronted with a chemical, they take it in, they then might be transformed in the body by enzymatic activities, all kinds of bioprocesses that can take place. Then it might be sequestered in hair, organs, fatty tissues, etc. So it's kind of put aside, so it's not as toxic within the body system. And of course, organisms will try to get rid of that chemical as quick as they can by making more water soluble if it's not water soluble, and so they can be excreted out of the body. We need to think of a higher level. So if organism is affected, and they might be affected in terms of, I said about mortality is the more obvious effects, more drastic effects, but might be affected in terms of growth, in terms of behavior, in terms of ability to reproduce. So it's a variety of effects that then might be observed at population level, at higher levels. So the organism, if it's not able to produce young, then they might disappear from the system. I put here life cycles because it's important to think about young, juveniles, embryos, etc. might be affected in a different way from adults as well as older organisms. James mentioned indicator species. It's important to distinguish, between what I'll call biomarkers in a minute and indicator species. They're different things. Biomarkers, well, I'll tell you a bit more about it, but indicator species basically tell us a bit about what's going on in the system. If you have predominance of resistant or sensitive species, as James has indicated in his examples. So what will happen then, the most affected or most sensitive species are the ones likely to be affected first by exposures to chemicals. They might disappear from the system, which might give opportunity to some species, the opportunistic species, the resistant species, to increase. And this is where we can use the indicator concept here to assess. And some species might be actually more important in keeping the community going and functioning in a proper way. So it is important to focus on that too. So this is kind of a diagrammatic way. What happens is an organism is faced with a chemical, they try to detoxify, there are natural ways of dealing with that. That might work, but they might not work, and the organism might die, or we might start actually see effects at higher level, of organism level, not at cell level or biochemical level. So the organism might then develop disease symptoms, for example, but then again, they might adapt at this level and all is good. If not, then you start seeing maybe changes at growth, reproduction, and so on, and performance, and then you might start seeing effects at population level, and eventually, as I said, it might cascade if one population die, then at community level, then you see effects. And this is where the link is with James and the other talk also on monitoring, where you can see differences in community structure. So biomarkers, as I said, we try to use biomarkers to assess effects of contaminants at a low level and at early stage. So measure physiological biochemical activity brought about by or as a result of a xenobiotic. What does that mean? Xeno means foreign, foreign in general, and here means a chemical that is extraneous to the system. Another definition, biological response to a chemical or chemicals that gives a measure of exposure, sometimes also of toxic effect. So we can have this to tell us that the organism has been exposed or affected, not necessarily one, but it can be both. So this is what happens. This is in a diagrammatic way. You have a chemical, you have effects at cellular level, tissue level, organ level, organism level, population, and eventually at community level, if the organism is not able to detoxify. But this is, there are ways, as I said before, dealing with the chemicals, so detoxification, storage, and potentially excretion. This is one of the common biomarkers that is used. This is a mechanistic process of activity of enzymes that is found in the liver of vertebrates or part of pancreas of invertebrates that basically deals with detoxification of non-water soluble chemicals, so dioxins, for example, but also PAHs. So it's very well known for dealing with those chemicals, sometimes quite successful. Those chemicals are biotransformed in the body, become water soluble, and they can be excreted. But it's not always possible that is as straightforward as that, so it can lead to further complications too. Nevertheless, this is a good biomarker to study to assess exposure to chemicals, so it's quite standard used in routine chronic toxicity tests. And what happens is that scientists use the activity of a specific group of enzymes that if they have an enhancement, that is that the organism is working hard to try to detoxify. All of us have this in our bodies, and if we drink, if we eat too much fatty food or too much alcohol or something like that, you would have this in action in your body anyway. So if you do monitoring of your liver activity, you can see that your liver is working faster to deal with these maybe extra food or extra types of compounds. So this is a kind of, as I said, standard procedure to look at the activity of certain functions in the body. But this got a specific link to exposures to lipophilic compounds, as I said. So what you have here is an example of that. So you have exposure to the chemical, you have activation of this system, detoxification, but the activation of this system, as I said, might not always lead to a straightforward detoxification. It might actually lead to generation of other compounds, which might act with the genetic compounds, the DNA and so on, the genetic system, which might actually lead to further complications. So this is the mixed functional, it's just another name. This is the kinds of things that we can study to assess if an organism has been exposed to some of these chemicals by studying, as I said, the activity of this system, but we also can study at genetic level. So I'm just giving you some examples of biomarkers that they've been used in this context. So I'll give you an example of a recent paper looking at fish populations in the North Sea. So they took samples from a variety of locations on haddock and cod, and they look also at body burdens of chemicals. So I did mention at the beginning, in order to study pollution, contamination, it's important to kind of look at chemical burdens or chemical availability in the field, as well as studies of activity in the body. So they took samples from a variety of locations. The location in the north of the North Sea, actually loads of platforms, central, not so many, and then in the south, where you have away from platforms. And they also took samples from an area around Iceland, where there were no platforms. They did find, for certain metabolites, they did find an increased level in the areas, so body burden of chemicals in the areas where there were more activity of oil platform activity. They also look at the MFO system, as I was telling you before, both for haddock and cod, and they actually see an increase in the areas where there were more platforms as opposed to the areas where there were less, but cod seemed to have a higher induction of that enzyme. They did do studies also on DNA damage, and so, you know, the DNA adducts, which is quite, again, a standard method to assess the formation of these adducts, the formation of damage of DNA on exposure to chemicals. And this is actually metabolites of the transformation of the chemicals, as I said before. And what they found is that, again, there was quite enhanced damage, DNA damage at a place where there was location of more activity. And with the Icelandic one, which in itself they found quite interesting, had less evidence of DNA damage as even some areas in the North Sea with very low incidence of oil exploration. So the conclusions from this is that the authors proposed there's a low level of pollution already being in chronic pollution being evidenced by the populations of fish within the North Sea as opposed to fish away, indicating that it's not just fish very close to the platforms, but it's the populations throughout the system. So this is kind of interesting aspect. This is scientific, it's not monitoring, but it's something that is important to keep tabs on because it's not something that can be seen like the year after or 6 months after. It's something that is seen in the longer term. Don't expect to read you this, but just say I wanted to tell you a little bit about the Sea Empress oil spill. It took place in '96. It was about 200, I think, 70,000 tons, more or less, that was actually spilled. It was February, so a time of low temperatures, not very high activity, but just on the verge of spring. There was dispersants applied at sea. There was cleanup also on shore because it was, they wanted to clean the beaches before the summer season, but also a protected area in terms of conservation interest. So there was quite a lot of activity taking place in terms of cleanup, in terms of using dispersants, but also on shore cleanup. And this is the area. This is actually close to where Richa is. So we got UK here, and this is the area where this took place. And this is actually where the tanker went aground, and this is the areas they were oiled. So one of our colleagues, Liz Dirinda, did quite a bit of work there by focusing on the mussels on the shore. So she carried out a immunotoxicology study using the methylacetylis mussel, and she's got a couple of publications on that. So these are the areas that we showed before, concentrated on 3 locations, and this is the control location which didn't have any oil coverage on the shore from that spillage. And it was quite well recorded that the body burdens of hydrocarbons on the mussels were quite high initially, but you could see they went down dramatically within the few months. By the summer, they were quite low, and this is the control area. Similarly here, this is phenanthrene. You can see, again, a standard decrease. She focused on 2 common biomarkers, one of them the superoxide generation and phagocytosis. They are common parameters of study when looking at the immunology of mussels, because they are important in defense mechanisms in the face of chemicals. So details of it here are not important, but similar to what I described before, the MFO system and DNA damage, some are important in terms of defense mechanisms, some are important in terms of the survival potential of the organism, because obviously if their DNA genetic material gets damaged, they might not survive. Well, in this case, as I said, their immune system became impaired, so that means that they were prone, if that was the case, to lead into disease generation. So what she noticed is that there were actually a decrease initially on the, and this is a superoxide generation, on exposure, well, after the spill, on the sites that were exposed, and significant differences from the control site. But there was also a natural lowering, as you can see here in the background. So some of these, this is important, as James mentioned, that we need to have good controls and distinguish from natural background concentrations. So it is possible that some of these responses are actually seasonally related anyway. So you need to bear this in mind. But then you can see then there was an increase again in later months. Similarly, this is the other biomarker they used, so there was a lower to start with on, you know, after the spill, and then again there was some kind of variability, but seems to kind of be, again, a degree of natural variability anyway. Very interestingly though, they look at body burdens of chemicals, specific chemicals, so such as the benzoylhalfopyrene, for example, but others also, and what they found, it was a pattern like this. Now, I look at this and I think, well, could it be because the oil could be transformed, or some of the oil could have been deposited and become available later, you know, from the oil spill, some of the components from the oil spill. But the authors actually proposed another interesting suggestion, which was they are derived from combustion. So actually they come from other sources. Some of the chemicals they observed, the hydrocarbons, they observe also in the burdens, and that seem to increase at later stages. So they're suggesting that this needs to be taken in consideration. Basically what I'm trying to say is that, like yesterday when I talk about the salt marsh, there are other pressures in the environment, there are other sources of potentially in this case hydrocarbons, but other chemicals, and this might lead to being the cause of the effect we are observing, and not, in this case, solely the spill. Okay, so I think there needs to be some kind of degree of caution when analyzing this data. Finally, they also look at another species. This was work actually done in laboratory. They look at another species of bivalve, and what they notice is actually the mussel was, although it's a standard organism for these kinds of studies, were actually more resistant than the other species of bivalve. So not all organisms react the same way when faced with a chemical. So we always need to be very careful when we compare species and conditions and situations. So there was severe immune suppression to start with, but that seemed to recover by the summer. So about 3 months after the spill, it was actually recovery to background level, so pretty fast. But as I said, they suggest there was variability, so there was a decline later in the year, and they link that to the availability of hydrocarbons such as benzoylhalfopyrene, which might have been generated from combustion processes. And this, as I said, is a proposal by the authors. Okay, so I think the overview from this oil spill, actually, just to go back, is that the recovery were actually much faster and better than it was actually feared, I think, by the organizations. And actually, one of our colleagues was heavily involved in advising government on the conservation aspects, and they actually come up with, him and colleagues, with a report which provides guidelines to deal with spills in sensitive areas. Very quickly, I want to refer to a colleague's work that James mentioned, Mark Hartles and his PhD student, work on chemicals from production of oil and gas associated activities. So they look at produced water, and they looked at some chemicals, corrosion inhibitor chemicals, and scale inhibitor chemicals, and they use a variety of biomarker techniques. These are the chemicals they look at, the quads, the azoline and the phosphate ester. And they look at actually concentrations of these chemicals in produced water, and what they did was they exposed mussels in the laboratory to concentrations which are realistic within these ranges that are found in produced water. And again, they look at some of the biomarkers I mentioned. They look at genetic damage, they look at oxidative stress. They were the key, yes, and lysosome stability, which I didn't mention. So they look at 3 types of biomarkers. I'm only showing one graph. It's just to basically to indicate that even at very low concentrations of the quads, there seem to be an indication of genetic damage of the mussels exposed. This is laboratory conditions. So it is interesting to see, and also, as I said, they did other biomarkers, and all of them indicated the same suggestions. Biomarkers are very useful to help us in the management. They are not part of monitoring necessarily, certainly not standard monitoring, but they help us to understand and to manage activities. And what I tried to say here, I gave you an example of general sampling from the field, an example after an oil spill, an example in the laboratory. So by complementing all of these, they can help us understand what happens in terms of the long-term chronic effects in the background of any chemical activity, or not just chemical, it could be physical, dredging, for example. So I think this complements well in terms of the scientists providing advice to both industrial practitioners as well as government in order to improve the management of such activity as this one.
¿Han tenido experiencias o trabajos conjuntos con las empresas prestadoras de los servicios de lodo, quienes también realizan pruebas de ecotoxicidad que les permitan validar, comparar y complementar los resultados de las pruebas de toxicidad?
Well, I actually did my PhD with BP. They sponsor me. So I think, and many university and scientists have worked very closely with industry. In many contexts, could it be looking at development and new or use of chemicals which are less toxic, looking at muds, looking at produced water in terms of effects in the laboratory, in the field. So I think there's good examples over the years. Some I'm familiar with from the UK, but I'm sure the same happened in other countries, where there's good collaboration in terms of progressing with the research, in this case in toxicity, at the bench, and that feeding into changes or, adapting to procedures that might be more environmentally friendly. So I think there's many examples in the literature and wide ranging.